Thank you. Is the mic on? Can you guys hear me okay? Firstly, thank you to everybody. You've all been just so delightful to talk to today. It's my last visit to Michigan was pre-pandemic, back in the before times, right? Before we all had to go home for 2 years. So it's really nice to be able to come back. You've got this brand new facility here. What a treat to be able, I understand this is probably one of only a few things you've had in this room. So anyways, it's helpful to and heartwarming to help you kind of inaugurate this new space for your new home. So thank you. I realized that my little laser pointer thing was running out of juice, so I'm just going to do the old-fashioned way and hit the space bar. So here's a little bit about me. As Ivo said, I've been around a while. I'm actually kind of from the Pacific Northwest in Seattle, and my family still lives there. I'll be traveling back and forth there actually next week. But I've been around. So after a postdoc at the NIH, went to Dartmouth College. Then I went to UCLA, where I worked very closely with Ivo on a number of different things. I moved across town to the University of Southern California. And then after spending far too much time in California, we needed a change of scenery, and the opportunity came up to help get involved with the founding of the new School of Data Science at the University of Virginia. It is one of the youngest schools in the UVA kind of collective of schools, but it's very exciting. I've had an opportunity to be part of building of centers and programs and institutes, but being part of a school, that's a whole different scale, and it's been really fun. I do encourage you to come and visit us. So one of the things I wanted to talk to you about today, here's kind of an outline of my talk, is I wanted to take you through this little journey that I'm going through with a student. I was asked to teach in our School of Data Science a course on the brain and data science. And I was assured that I was going to have 12 graduate students, PhD students, who were going to come and take this class. So I built up a whole curriculum. And then on the very first day of class, I had 3 students, and 2 students dropped. So I had one guy left. Ethan Nelson is his name. And so I threw my syllabus out the window, right? We're not going to need this. And he and I just started talking about neural networks and about their role in data science and about what they may mean in biological science and why they're sort of at odds with each other. So I want to take you on this journey about how we've arrived at this idea of these, what we call digital neural organoids, which are a computational thing. So if you've come here to learn about organoids that you grow in a lab in a petri dish, I am not your guy, but I'm hoping that you'll see the connection and that you'll go, oh yeah, that makes perfect sense. That's exactly what you would call these. So bear with me a little bit, suspend your disbelief. We'll get there, but I'm going to try and talk about some of the inspirations for this in classic neurophysiology, neural networks, actual biological organoid technologies with as little as I know about it, things in optogenetics that help to inspire how we might stimulate these things and make them work, as well as the larger picture of human brain imaging, which I've been doing for 34 years. And then talk about this organoid model and some of the properties that it has and why we're intrigued by it. When I get to the end, you're going to be woefully disappointed because I don't have an answer for you yet. Invite me back in a year and I'll tell you how it worked out, okay? So that's what we're going to try to do. So I spend a lot of time, have spent a lot of time since, for years, since I've been doing human neuroimaging, trying to understand the structure, the function, and the connectivity of the brain across multiple different scales, across multiple different disease types. It's basically what I do. I have published extensively in autism spectrum disorder, in Parkinson's disease, Alzheimer's, traumatic brain injury, and trying to understand the brain in its form and its function in health and disease is what I do. And when I joined the University of Virginia, I realized that I had to kind of do this in 2 different ways. I have a joint position, so half of me is based in the School of Arts and Sciences in the psychology department. That's actually my academic home. My other half of me is in the School of Data Science. When I'm in arts and sciences, I kind of have to talk about the brain that looks like this, right? Talk about neural networks in terms of structural connectivity, functional correlations over space and over time using neuroimaging. And we've done various things of looking across different spatial and temporal scales, looking across these topological frameworks and whatnot. That's how we kind of characterize and understand the brain in my neuroimaging life. On the other hand, when I walk down the street and I walk into the School of Data Science, I'm often asked to talk about neural networks that look like this. This is probably not unfamiliar to most of you. This is kind of what we think about when we talk about neural networks. This is a very basic form. But you'll notice that this doesn't really look very much like this. And yet when we talk about this, or rather when certain people talk about this, they're saying, oh, we're basically modeling how the brain works. And like, my feeling is that's not entirely true. So when young Ethan and I realized we were the only people who were going to be involved in this class I was supposed to teach, we kind of started to have some discussions about that. We went through this arc of asking questions about how can we take pieces from this and infuse them into this, which gives us a more biologically looking like thing. One of the key things we realized was that these, when you run these, they happen instantaneously. The speed of light, right? You put in your inputs, the math happens, the matrix algebra happens, but you get your answer right away. The brain takes time to send those signals from one side of the brain to the other. It's fast, but it's not instantaneous. We wanted to capture some of that. There's some other temporal dynamics and spatial dynamics we wanted to capture that these don't capture, but certainly is captured by this side. So it's like, how can we bring these together? So this was kind of our arc, starting with foundational neuroscience. Many of you are, how many neuroscientists do we have here? I mean, you don't even look convinced you're a neuroscience. You're sort of like, maybe. Want to see like this, right? So many of you may be very familiar of the 6-layer model of the cerebral cortex, the different kind of the whole zoological framework of different neural cells, how they arrange themselves, these bands of biology that cut across within a layer, connecting the layer to cells within its same layer, as well as these things which cut through the cortex, going out to the rest of the central nervous system and connecting one part of the brain to another part of the brain. Some of these, the individual cells that participate in that, you can isolate them. And that is like a lot of what neuroscience is trying to isolate those cells, understand their properties that we don't necessarily care about so much in neural networks. We kind of worry about the whole system and its ability to classify things. Neurons don't care, right? Neurons care that their neighbor fired so that they'll fire too, if it reaches a particular level. But this is something which was inspiring to us, that neural networks, they're not really inspired by this structure, this notion of being able to communicate within a layer as well as across layers and leverage those signals as they move through that system. Likewise, these signals have direction. They go somewhere. And Ramon E. Cajal, the famous neuroanatomist, actually points that out in some of his diagrams that he created. You'll notice that he's got these pictures of these neurons, but if you look close enough, you'll notice that there's little arrows here, right? So here's one, indicating that there's a direction of flow of information through this structure. Can anybody tell me what that structure is? This is the hippocampus. What is the part of your brain which is most susceptible in Alzheimer's disease? The hippocampus, right? So you have hippocampal damage in Alzheimer's disease, the flow of information through the hippocampus gets affected. So that information is not only going along lamina, it's going across lamina, and it has a direction of flow through the brain. This was another thing. Now, in neural networks, there's a whole bunch of different configurations. So we understand that. But one of the things that people build with these, when they build them, they give them the neural-like property of they have these activation functions. Many of you are familiar, the ReLU function, the rectilinear kind of activation function, that if the activity at a node in one of these networks is above some threshold, that thing is going to fire, right? And so you have a whole family of these that people have utilized. The ReLU function, this rectified linear unit right here, is the most kind of famous one. But these things, especially this one, are really the only thing that's neural about the neural network, right? Otherwise, it's just a connected graph. It's with that little activation function that neurons do that give it that neural network title. The fact that we've given it that neural network title has sort of reified all this into thinking that these things are real or that they're emulating the brain. They sort of do brain-like things, but really you're just deciding that you're going to use this activation function or any of these others and produce a neural network. To me, that is necessary, but it is by no means sufficient. And that worried me, that no matter how you configure this, choosing of just one of these functions does not make it a neural network. So that was another thing we talked about. If you look closely at how axons are constructed, right, what will you notice? You'll notice that they have a axonal pathway here that comes out of the cell. The Schwann cells or oligodendrocytes in the central nervous system are the insulating material that wraps around them to produce these little curly Q looking guys that provide insulation, they provide nourishment, they basically help to propagate the signals down the axon through something called saltatory conduction. Saltatory, saltare is Italian for hopping, that the signal looks like it hops down the neuron. That's essential. And as it turns out, the speed of that saltatory conduction is a function of how myelinated that particular axon is. So speed is controlled by the relationship of the myelin sheath to the diameter of the axonal membrane. Here's kind of an electron micrograph of what that may look like in close up. So you can see the myelin sheaths are wrapped around those axons in this fasciculi. I think it came from a mouse. And you can kind of see what that structure looks like. This is something which neural networks do not respect and do not take into account. So one other thing that we were worried about. Another thing is that those signals take time. They're attenuated. They are a traveling wave. So in a neural network, it happens instantaneously, right? There's no traveling necessarily. It's matrix algebra. We wanted to capture this as well. So of all these things, we went back to some foundational neuroanatomy that got Eccles, Hodgkin, and Huxley the Nobel Prize back in the day, where they worked out this thing called the cable equation. This is the fundamental equation that you usually learn about in any sort of neurophysiological textbook. Within about the first 20 pages of that textbook, you will see a version of this equation. You will notice that it's a partial differential equation. It takes into account space and it takes into account time. There's some electrical parameters as well, which you'll see a little more detailed in a second, which is basically trying to describe the current as it flows down the axon. This was something we wanted to capture as well. What's interesting about this particular equation is it's a simplification of another equation, which is called the telegrapher's equation. The telegrapher's equation is a little more expansive. It actually involves electrical fields and magnetic fields, and is actually kind of where its origin comes from, where it sounds like. It was transatlantic telephone cables, and it was Oliver Heaviside who kind of worked out this fundamental form of these two paired equations. But actually, Lord Kelvin was the first person to start considering this, and is actually born out of James Clerk Maxwell's fundamental equations, which you can all see if you go buy a coffee mug or a T-shirt. They have them on there now. You can go buy those. But this goes back to basically the turn of the 1900s and working out the signal properties of transatlantic telephone cables. Because back in those days, it would take like 20 minutes to send one character from one side of the Atlantic to the other. And they wanted to figure that out. So they came up with this equation. The simplification of this to turn it into the cable equation, which is used in neuronal signal propagation, is to assume that no induction exists. And so if you do that, this whole thing collapses and you get the cable equation. And that was Hodgkin and Huxley and Eccles' big insight. But all these electrical parameters are part of how this thing works. That's something else that we wanted to try to capture. One other thing is I mentioned sort of in passing, this notion of that myelination is super important. Axons which are unmyelinated, the signal will travel at the square root of the diameter of that axon. So these are in the level of the cortex, not in white matter, but in the cortex, they're unmyelinated. So in moving across it within a particular lamina, at a rate which is proportional to the square root of the diameter. In white matter, you're going to get them, it will travel proportionally, but also in a little more complicated way as a function of the ratio of the axon diameter to the myelination diameter. This is classically referred to as the G ratio. And you can look at this when you look at, say, electron microscopy of pretty much any neural system. You can see the wrappings of that oligodendrocyte wrapping around the axon, providing that insulation. And because of that, that signal can travel faster. And I'll show you the kind of equation of motion for that here in a second. But it's as people have done various experiments, they've demonstrated that that's the case. And then white matter tractography in neuroimaging. So many of you are probably familiar with functional magnetic resonance imaging, where you're looking at, say, signal activation over time in response to a stimulus, a cognitive stimulus or visual, auditory, whatever stimulus. This is looking at the white matter fiber pathways in the brain using something called diffusion weighted imaging. Manipulating the magnetic field gradients across the person's brain who you're scanning. You can then go and identify the preferred directions of water molecule diffusivity, and by indirection, you can get an analogy to the directions of the white matter fiber pathways. And so then with a little bit of math, you can create images which look like this of white matter fiber pathways. Now you're probably saying, but Jack, there's nothing white about these images. They're red, blue, and green. In these images, the convention is if anything is traveling left to right, they're going to color it red. Anything that's traveling anterior to posterior in the brain, they're going to color it green. Anything that's going inferior to superior, they're going to color blue. And so those help in this image to give you a sense of which way those fibers are oriented. This doesn't tell you about direction of travel, but it affords you the ability to go and break things up into various fasciculi and then do graph statistics on them. So this is another very powerful technique that we use in human neuroimaging. Touch basing on conduction velocity again, here's Dr. Rushton, who was a contemporary of Hodgkin and Huxley, and he realized that if you knew that G ratio, that the velocity of the signal, basically the not the bandwidth, but the wave length of the signal, if you will, is proportional to the internodal distance between the nodes. Many of you may remember that these the nodes in between these oligodendrocyte where the myelination happens there's a distance between them, and that's where the ionic transfer happens, is also proportional to this, is proportional to all of this business. And this is how you can go and find that the optimal value of this G ratio turns out to be approximately 0.606, which is 1 over the square root of E, as it turns out. This is born out of the electrical properties of how myelinated axons actually work. So this is kind of nice. If you know that you have an optimal G, that's the maximum speed that you can get. If you have more myelination than you need, that means your G ratio goes down and your velocity goes down. If you have too little myelination, your G ratio goes up and your signal falls off again. So this is kind of the optimal peak, is where you have the amount of myelin to the amount of axon you have is in this 0.606 ratio. Your signals are traveling as fast as they can. And that little movie that played was just kind of some simulation of some what G ratio is. Turns out we can actually estimate this G ratio at every point in the brain using neuroimaging. So using that methodology of water molecule diffusion properties with a little bit of magic and a little bit of combining that information with structural related information, we can get an estimate of the G ratio at every point in the brain. And one thing you'll realize is that it's highlighting the white matter. The G ratio is going to be the strongest where there is white matter, and that is going to be very helpful. If we look at this T1, T2 ratio, this is T1 as a property of the magnetic signal that we get. You have two parameters, T1 and T2. The ratio of those give you an idea of the overall density of the particular tissue at those locations. And then with a little bit of a transform, sorry that my little thing at the bottom here is in the way, gives you a map of that conduction velocity. I think I can show you another way we can do this. So there's a workflow that takes the data from the scanner for the same individual, multiple modalities, and can turn them into this G ratio, which can basically be converted to a conduction velocity indicating that things like the corpus callosum here, that's an information superhighway, right? Signals are traveling really fast. If you get out here into some of these other areas, signals are traveling a little bit slower, probably at the rate proportional to the square root of the diameter of axons in those areas. The splenium of the corpus callosum, the extreme and external capsules of white matter, very, very high throughput. You might think of these as the highway systems, whereas some of these others are more of the, off-roads, the gravel country roads, if you will, of information flow. But using this, we've been able to apply this to find conduction velocity differences in people who have autism spectrum disorder. We published that in PLOS One a couple years ago. We've done work with Parkinson's disease, and we've also done some other work in developing babies. And so this is a very rich area from our neuroimaging perspective, but we also wanted to capitalize on this as well. Functional MRI is a process where you go through and you present stimuli to people in the scanner, and you look at a thing called the blood oxygenation level dependent effect. This is the fortuitous property that deoxygenated hemoglobin is slightly more paramagnetic than oxygenated hemoglobin. So where cells are using more oxygen, you would expect to see more signal in those areas. So this was a study we did a number of years ago with my colleague Peter Giannata, who's now at UC Davis. We published this in Science now a million years ago, but where we were presenting musical tones to people in the scanner. So we had some special headphones made up to overcome the sound of the scanner, and we're projecting basically frequency information based upon Western tonal music into these things. We're basically playing them a little tune, and the little tune would change its key, and as it did, it would migrate this little profile, spectral profile, through a bunch of different keys, and we were able to then go and map that to different locations in the brain. Clearly, auditory cortex is lighting up, but also we were able to see areas of the kind of medial frontal cortex as well. And so we were able to produce activation based upon the underlying patterns. We were able to associate these different spectral patterns with these different areas of the brain. This was kind of really cool. Maybe that's why it got published in Science. So at any rate, that is a huge, big thing. The other thing that kind of inspired us, so you'll see that there's multiple threads here. We're trying to all bring together. So this is a bit of an intellectual challenge, is the use of optogenetics. So many of you may be doing some work in optogenetics. I'm not sure. I'm not really an expert on this, but you basically have these little molds that you can extract, algaes, which you can extract out the fundamental channel rhodopsin chromosomes genes, and you can splice them into either frogs or mice. And then when you shine light, usually using a fiber optic on particular areas of the brain, you can get those cells to fire. And when you can get those cells to fire, you can control the animal, right? So you could go and have them actually run a maze. You can produce different kind of frequencies. You can basically get them to do all sorts of things by firing light at their neurons. This is really cool. So in thinking about all this stuff, we're trying to think about how can we put all this stuff together? The other thing we wanted to be able to do is capitalize on this notion of how the brain seems to organize itself. There's a methodology called independent components analysis, which I have all sorts of problems with, but it is one of the ways in which many of you may have heard of the default mode network in the brain. This is basically what's going on when your brain is left to its own devices and you're laying in the scanner thinking about everything else but what the experimentalists have asked you to do. The default mode looks a little bit like this, but there's these other modes that we have sort of named, these things like the executive control network, the auditory, the salience, sensory motor, visual lateral, visual occipital, and visual medial. All these different networks are things that are really interesting to extract from the brain. People have turned this into whole research, built their careers around these things using some very simple linear algebra type things that you can do. I worry about these a little bit. I'll tell you that maybe another time. But this is a way that people process that functional data to basically look at the brain networks that are underlying the function. So then finally, one of the things which has emerged recently is some colleagues that I have in Australia have started to look at what they call these eigenmodes. So instead of using ICA, they're fitting, we're basically deriving partial differential equations on the surface of the cortex to get at these traveling waves of activity, right? So things like the ICA result gives you a static map. It says, here's the network. But on the other hand, this is probably a little more akin to how the brain's working. You've got these traveling waves of activity traveling over the cortex through the white matter, getting from one part of the brain to the other. They've extensively explored this to produce these different kind of eigenmodes to basically solve this thing called the Helmholtz equation, and then basically they can play these things out over time.
Get this with EEG? I mean,
This is with fMRI data.
how
So they're using fMRI data.
you can go inside.
Yeah, and you can, of course, you can also try to do this stuff with EEG data as well, but here their application was to use fMRI data as the thing. And they've actually gone through a number of, they mined a big database, speaking of like big databases that are available. They went through, I think it was the UK Biobank data set, and they pulled out tasks which were associated with social cognition, motor cognition, gambling, working memory, language, emotion, and relational cognitive processes, and did these, and they were able to produce these different patterns of waves that were stimulus specific. This to me is really, really cool because it's not just that time dimension, it's the spatial dimension as well. So bringing all these things together was kind of fun. Here's just a little bit of the work that we did. This is some work from my postdoctoral fellow, Ben Newman, looking at these differences in conduction velocity. You can look at G ratios in people who have an autism spectrum disorder diagnosis and typically developing controls. And when we look at the conduction velocities, you see that the people who have the autism diagnosis are slightly less fast than those who are typically developing controls. And we can map this pretty much in any region of the brain. And again, this was published in PLOS One and has been very exciting for us. So my student Ethan and I started wondering, how can you bring all this stuff together, especially this like last bit with these partial differential equations, because that velocity term, the speed factor, is a property of those waves, if you will. You can tune that. So if you turn it up, make the wave travel faster. You can turn it down, the wave travels slower. So we thought about what you would do if you had this little G ratio function that governed the wave, and then we made a wave equation which would do this. So I got to play these things again just so you can see. A couple of key things about partial differential equations. How many mathematicians do we have? What do you know about PDEs? Yeah?
I taught him.
Yeah, that's right. Well, one of the key things is there's a kind of a general form, and then you have to make a decision about the boundary conditions, right? Once you've made a decision about the boundary conditions, usually a lot of stuff falls away and you get a simpler kind of a model. So here is like, whoops, excuse me. Hey. Oh no, I broke it. No, I'm sorry about that. Hang on. We'll get this back here. Here we go. Sorry. Okay. So here is where you have a non-reflecting boundary. Here's where you have a reflecting boundary. What's going to happen when you have the reflection? There you're going to go. It's going to reflect, right? So we don't want that. We want something with a non-reflective boundary. And so anyway, we worked out the math to make sure that all of this works. But what we wanted to do was in this axon, we want to have an equation of that traveling motion through the axon to be able to produce this with a non-reflective boundary so it would travel down the axon and not bounce back. So we had to think about these things. This is about as mathy as this gets. There might be one or two more, but I promised I wouldn't show too many math equations. Oops, what's going on here? Oh, yes. So there's all these different things, and our insight was kind of twofold. One, we thought many times you're looking at neural networks which look like this, which sort of read left to right. And our thought was, well, wait a minute, what if you took that neural network, and what if you could, instead of it looking like this, what if you made these nodes movable in space, right? You might tie them together because they're in the same layer, but you make them movable. And then what happens if you take it and you wrap it around a circle? Well, if you can wrap it around a circle this way, you can also wrap it around a circle this way, and you get a sphere. So here is like a cross-sectional version of wrapping one of these guys kind of around its own axis. But then these connections between the nodes in this network, we gave a G ratio. we govern the speed. And things that are in the same layer will connect without, they won't be myelinated, but anything that's long range will be governed by our telegrapher's equations and be governed by that speed coefficient that we can produce, right? So we can change the speed of these things. So we thought, let's turn that into something. So we did, but we were inspired also, once we had this spherical shape, we're very familiar with work, and I know that there's lots of people doing this. Oops, this thing's in the way, apologize. I think that can be hidden here. There we go, get rid of that. Many of you are working in this the space of these actual biological organoids, which are three-dimensional depictions of pluripotent stem cells. You know, put them in the oven, you bake them at 350, and you make some other, there's a recipe, right? And you get these things that are three-dimensional. They have that kind of near spherical shape sometimes, and they're really quite beautiful to look at. Because we started thinking about these neural networks as spheres, it was a pretty easy leap to just start calling them organoids. And so these are digital neural organoids, not real biological organoids. And we do not mean to suggest that these are digital twins of organoids. They may have elements of that give you that flavor, but that is not our intention. But so we end up with these three-dimensional structures which look a lot like a pluripotent stem cell based organoid. And they actually, this is a depiction of what one looks like where I've sort of made the upper hemisphere of this transparent. You'll see that it has a very rich structure, right? You've got this outer layer here, which is the blue ones, then there's a lighter blue one, a green one, and a yellow one, and then at the core of this is a set of red nodes. So imagine a neural network that you've wrapped around this way, and you've wrapped around this way, and now it's in a ball. Notice the degree of connectivity that exists within a layer. Every node is immediately connected to its immediate 6 neighbors. We use a Fibonacci mesh to create these, and so everything has the same number of connections, excuse me, and so they have 6 neighbors. Everything is connected to its neighbor, as well as connecting through from layer to layer, and then the pooling layer that we've created actually connects to its antipodal node so that it's sending signals through a medullated fiber all the way across the organoid to the other side. This has some cool properties. It's a three-dimensional configuration of cells. There are these shells or cortical layers that produce a cortex. It has a white matter architecture in the middle of it. You have this kind of spatial connectiveness to it, which we think is really interesting. Basically, voltages travel through this using Ohm's law, using Kirchhoff's law, et cetera, all governed by this underlying telegrapher's equation. And it has a number of other different things, Hodgkin and Huxley-based modeling, thicker long-range connections are modulated, as I said. And again, this is the idea is to build a structure which has the dynamics associated with it, which we think is something the way the brain takes advantage of those dynamics. Current neural networks don't do this. So this is just a depiction of what it looks like, but it really does look like that when it gets rendered. And here's some other notions. Again, nested Fibonacci meshes. And then here's where I take away the connectivity so you can see the construction of the nodes. And then this is, so again, the blue one is the input layer. There's a convolutional number 1 layer, convolutional 2 layer, and a pooling layer. And then there's this output layer. This output layer is kind of where all the action happens. And I'll show you some videos of what this looks like. Here's just more detail about the connected structure. And you can see how densely connected it is. But it's actually pretty simple. The degree of connectivity for any particular node is 6, which is not all that much compared to other things in the brain. So we can take advantage of some of that and produce some really interesting dynamics. Now, how do we stimulate this? We use a graphical programming trick called UV mapping, where we can take, basically you can do this but in any graphics software that you may be familiar with. Who uses Blender for stuff? Yeah, Blender will take an image and map it onto any three-dimensional object using UV mapping. So we're using the same trick. This is like our virtual optogenetic approach. We're projecting an image, like for example, we've also played around with the MNIST database set of characters. And so for example, here's an organoid structure, and we've painted an 8 to the surface of this. Everywhere where there's basically a part of the 8 is going to stimulate those nodes, right? We can change the size of that, the extent of that degree of stimulation. We can move it around in space. We can twist it. We can move it. And so that can be very helpful. And this uses this mathematical technique of UV mapping, which comes from graphical software. And we've also played around with a variety of different stimulus types. I mentioned those, the MNIST database. We're working a little bit with those. But the one we're going to focus on today is where we present basically faces of dice. So we're also playing around with looking at 1, 2, 3, the actual numbers or the words that describe those. But I'm going to show you some of the results that's come from looking at these guys, where we're taking that image, we're using the UV mapping, we're projecting it to that input layer, and in so doing, we're stimulating this organoid to do stuff. So when we do that, here's an example of when we project a particular stimuli onto the surface of the organoid. Here is the time courses that are kind of running down here for the input layer, for the convolutional layer, pooling layer, and then the output. This is a version of the output layer. And one thing you'll notice, and some apologies, it's a little herky-jerky here. I think my computer is just overwhelmed with trying to project to too many things, is that you get that traveling wave phenomenon that we were trying to capture, right? This is really cool because we're able to project an image and get this pattern. Now you'll notice that it takes time for that signal to get to, that's the output layer. So when it be here, it's at the input layer, it's the first thing it sees, right? But then it travels this way. So it takes a little bit of a delay, a little bit more of a delay, a little bit more of a delay till it finally reaches here. So time is elapsed until I see that part. If we go and do this and look at a kind of a principal component analysis approach to this, we go and we get this really curly Q kind of trajectory of the signals as they move through space. This is kind of neat because if you explore this a little bit more and you look at how the different wave types can be generated when you give it a 1, a 2, or a 3-faced dice, these are going to need me to, oh, here we go. Okay, so here's a 1. So kind of keep your eye on the pattern there. So that's a 1. That's pretty simple. Now when the next one starts, I'm just going to force that to go. Give it a second here. So this is now 2 dots that are being presented. Hang on, it'll get there. Keep going. Sorry, it's taking a little bit of time. My computer's so slow. Here we go. There's 2 dots. I'm going to start this one just so you see what the 3rd dot looks like. The wave pattern that you get is more complicated. There's more nuance. There's a spatial separation. There's wave interference, and so they're canceling each other out or they're reinforcing each other. That's something you would hope to see in a wave kind of driven system. And you get these very, you know, we colored this, we use the Inferno color scale. I'm a big fan of that one, to basically produce these. So it's really kind of fun. And if you look at these things in kind of time, we've rank ordered them by the first arrival time of one of the action potentials that's being modeled by that telegrapher's equation. And if you rank order them, you see you have this initial wave passage, but you have all these other little transients going on. So if you watch these things long enough, they start to become very hypnotic. You start to see vorticities in the system, and you start to see some of those things which are much like a reaction diffusion kind of equation. We didn't build it in. It's just happening, which is really kind of neat on the basis of how things are connected internally. So kind of neat. Oops. There we go. Another way we can do this is if many of you are familiar with a Mercator projection, you know, that image of the flattened Earth, right? So if we present these this way, it really helps to showcase the clever dynamics that are going on here. And this is going to run a little bit slower just so that we can kind of watch things evolve here. The way these signals evolve, I think this is where the action is, because how they're actually spatially arranged, how they're evolving over time, how they spread, you'll see lots of different dynamics going on if you play these long enough, where things start to go into some of these upper latitudes and they start to actually become much more spread out, much more kind kind of dynamic. And if you wait for these things to kind of settle into some of those transient dynamics, it gets even more crazy. So this is some of the things we're able to produce with our simple little organoids that we've created. Let me see if I can advance this properly here. There we go. If you play these things out long enough, that notion of that kind of almost like chaotic trajectory is really borne out when you look at these things in kind of a principal components over time space. You can see that we basically turn the stimulus on. We showed it a picture of, I'm not exactly sure. Oh, here we are. All 6 different dice faces. We just turned them on and left them on for a while to watch what happens to the dynamics. And it gets into this kind of cycle, but that cycle attenuates itself over time. So this whole kind of nonlinear dynamic structure is something which is just an emergent property of these things, which we think is really cool. Here's looking down on it. So same plot, but you're looking down on it. Same plot, you're looking down on it. You can see these different trajectories it's taken based upon which dice face happened to be shown. So again, kind of neat. So we're excited about this. Now, we're trying to also do something that's different. Instead of just looking at these things and like having all these time series, we've asked the question, how can we change the morphometry of the organoid based upon these signal characteristics? So what we do is we take that output node, it's all, or the output set of nodes, it's always spherical. So it's always a sphere. We don't change that one, but we look at every latitude and longitude, every azimuth and elevation, and we ask the question, can we fit this equation, which is called the spherical harmonic equation? It shows up all the time in physics, in astrosciences, you name it, it shows up everywhere, where you can, it's like basically a Fourier transform on the surface of the sphere. So we can go through and we can, here's like a characterization of looking at a dice face 1, I believe, where you get these different L modes, and you get, basically can do these at different time steps. So you can watch the dynamics play out as a Fourier transform on the surface of a sphere. By taking the frequency, the phase, and the magnitude information, we can feed that back into the system, and we can deform the organoid's shape. So any learning that's going to go on with this is not going to be carrying around this bunch of weights like a neural network, traditional neural network would be. It's going to be by changing its shape. We're going to optimize geometry to take advantage of time. And we're going to take advantage of time to optimize the geometry. When we had this sort of revelation, our heads kind of exploded. This was really kind of exciting. So here are a couple of different, the spectral characteristics. Looking at the power spectrum over the multipole moment, or basically what's called this L term, looking over those different things up to an L of 50. And you'll see that there's this little kind of collection of frequencies down here in these relatively lower multipoles. But when you get up to these higher multipoles, this is where that information starts to distinguish itself between what you stimulated it with, whether it was a 6, whether it was a 4, whether it was a 2, etc. And you can use that information to reconstruct the waveform, but you can also use that to modulate the waveform. So we've started doing this too. So here's just more of a complete spectrum of looking at a dice face. One, apologies about the resolution on this. It's not as clean as I'd like, but if this is a dice face 1, if I hit a space bar, this is a dice face 2. You'll notice that all those dynamical changes are reflective through this decomposition of the organoid's output surface. And then here we can actually break things down and look at the individual contributions of each one of those spherical harmonics to the overall signal. And this is looks better with the lights off, I think, but you'll be able to see this. This is an L of 1, so basically one component. Here's 3, 5, 7, and 25. Each contributing to the underlying dynamics over the surface of the organoid. So these are kind of cool. This plays out over time, but it's running a little slow. How are we doing here? I'm almost to the end. And here is how we're going to go about transforming these. So we can go and create a length. We have basically a distance, basically the length of that fiber pathway. We know its velocity. If we divide the length by the velocity, we're going to get a time. If we compare that to some optimal time that we will have found. We can use that to build our cost function. We can also use it to build our updating function. And by that, we can stretch the organoid. Likewise, we could do this in terms of phase angles as well. And that's kind of what we've done here. Here's a simple example of this where we can change things horizontally. So let's look at this real quick. So you can stretch it in each axis. Now, these are the two simplest things. We can actually do much more complicated things than that. But if we just go and calculate those, we can demonstrate to ourselves that we can adjust the phase difference. That is, we can adjust the timing of signal arrival times so that they all converge in phase at the same time at the output layer. So they're not chaotic anymore. Now we can actually start to shape this thing, and we can actually build it so it optimizes those signal arrival times. This is really kind of a powerful, powerful thing that I'm unaware of anyone really playing around with in neural networks the way we kind of traditionally do them. So this is just an example of some experiments that we did where we're systematically manipulating how much we could stretch the axes in these different directions to produce a modulated signal. And here's just some summary statistics which illustrate that when you change the axes or you stretch it out, you're basically changing the arrival times. You're looking at different frequency spectra. You're able to manipulate that and control this information as it flows through these organoids. So here's our next steps, which we haven't done. And this is where my whole talk comes to a crashing halt because I don't have the answer for you about whether or not the organoids can learn yet. But again, invite me back in a year and I'll give you an update. But the idea is to start with our pluripotent naive organoid, to have it go through this process of changing the various shapes, changing the various axes, using that frequency information to manipulate it, right? And change that shape and find that idealized place so that we end up with an organoid which is not round anymore. It now has this very dynamic topology to it. From that, we can do all sorts of graph statistics. We can ask about this mean path length. We can ask about small worldness. We can ask about all sorts of community structure that's evolved. So these kinds of things, which you may all do now in graph theoretical applications, we should be able to do here too. We should be able to look at a number of different properties, optimizing them from their relatively untrained state into something which is much more robust, narrowed, focused, and gives us the kind of performance characteristics we're really interested in, and then be able to apply those learning rules to be able to show that this thing can tell the difference between a 1 and a 6 and a 2 and a 4, and pictures of 8s and pictures of 6s and that sort of thing. So we're very close. I expect actually that we'll have some of this in place next week, but we're going to go through a little process because, as I think I mentioned, trying to apply conventional neural network learning rules in 3D like this, it doesn't work. I'll save you the effort. Don't do it because it will waste your time. It doesn't work. So as we're coming up with this is kind of how we've arrived here, my student Ethan and I. We've drawn from these multiple different things to make something that's a neural network much more neurally inspired, much more neurally based. Really trying to, here we are. Trying to develop these neural network systems, which are much more neural-like, as I said. Blend the foundational neuroscience into these things, give them those properties, right? Create this little model system that basically is a digital petri dish. Once we've created these and teach them how to learn, one of the things we want to do is give it a stroke. We want to say, give it multiple sclerosis. What's going to happen, right? Is it going to still be able to do what we expect it to do? Are there some of these recent concepts of microstructural modeling? Some of you may be working in this space, certainly as we're doing it in human brain imaging. Bring those ideas into this system so that we're creating this little microcosm that we can attempt to manipulate and understand before we attempt something like a full-scale digital twin of a whole brain, which would be a very computationally challenging effort. These are three-dimensional. They're no longer this dimensionless, left to right red thing. They're three-dimensional. This gives them a place. You can imagine, for those of you who do organoid kind of related work, you're very familiar with assembloids, right? What happens if you go and you build a number of these and then you link them together to create a digital neural assembloid? There's a lot of potential here that is really kind of exciting. They're a very useful analogy or model. We're not trying to make a digital twin of an actual organoid, but we're inspired by that space that it occupies, the shape and the structure that organoids happen to have. It's a very convenient analogy. And we're really looking at voltages over space and time. This, to my mind, brought, at least into my thinking, a lot of the work that our good friend Dr. Dienov does with his space-time modeling, where you're looking at these signals which evolve over time. They're complex, they have phase, they have magnitude, and they are something that's very, very rich, have a lot of implications for how these systems might work. One thing which kind of is born out of this is if you take the number of nodes that you have and say that scales to infinity, you get some very interesting equations which really kind of speak to space and time and how we understand it at the level of the brain. The neuron disappears. It's all part of optimizing geometry and time. And this would be a perfect example for playing with some of your methodologies. We've created an excitable medium, much like a neuron itself, where you stimulate the bilipid layer and you produce basically this traveling waveform. We've done that, at least in silico here, as our little toy problem. And some of these behaviors are certainly reminiscent or duplicate or kind of imitate traveling waves, spiral waveforms, vorticity, very highly complex dynamics. And I'm really excited to play around with that. And this linkage between geometry, time, and waves of activity are really going to be where we do any learning. We're not going to learn on the basis of it's a cat, it's a dog. We're going to use the wave information as the thing we learn against. And this, again, is something which I'm not familiar with in traditional neural networks anyway. And really, this is an experimental test bed or a petri dish for us to go in, create these things, teach them to learn, and then lesion them, find out what happens to them if we break them. So we're very excited about this. I want to basically acknowledge this is a lot of Ethan Nelson's work. He is a third-year PhD student. He went to the Network Neurophysiology Conference in Lake Como last summer, and he got the best presentation award for this work. My colleague Ben Newman is really somebody who's really helped us with creating the foundations of this in terms of conduction velocity. And Zach Jacobs is doing a lot of the work in our autism area. And with fingers crossed, he's going to graduate with his PhD in May. And so these are the guys who really helped. And of course, we did this at the University of Virginia's College of Arts and Sciences and School of Data Science, and with the help of research computing, where we take advantage of their GPU capabilities to be able to run these things. And I would like to invite all of you to reciprocate my visit here by coming to Charlottesville and hanging out with Thomas Jefferson and I, and we'll show you a good time in our School of Data Science. I think I talked really fast, I'm sorry, but I hope we have some time for questions. Thank you, everybody.
Questions and comments?
Yes, sir.
So, based on this spherical structure, it seems like if you're keeping the number of connections constant at 6, as you said, for each layer that you add, you're going to be adding exponentially more and more neurons. And so do you in the future plan on doing some sort of process similar to pruning that happens in early development as part of your training? And then I guess my second question is glial cells also play a big role in like modulating conductance, the G value that you mentioned.
So for the first part of your question, yes, absolutely. So we've thought about like Hebbian sort of rules, like certainly any connection which is underused or underutilized, basically set to 0. It doesn't exist anymore. So we could remove it, right, computationally. On the other hand, if it looks like there's a node which is just being inundated, we might actually spawn another connection. So create one where one didn't exist. So we've thought about doing that. As far as glial cells, we haven't thought about that yet. That's a couple steps away. That might be version 2.0, but that would be another thing that you could do to have, more of a support structure throughout the system. So we'll be looking at that sort of thing too in the future.
Yeah. Thank you so much. This was amazing.
Oh, thank you. Appreciate that. Yes, sir. Way in the back.
I'll just say it. All right, we're the Zoom.
For the Zoom people.
Oh, Okay.
Okay, yes.
All right. Okay. Great presentation. Thank you so much.
Thank you.
I just wanted to ask, like, how loyal are we to the Ohm's law according to, like, the speed that the thing travels? Because to me, it sounds like pretty expensive. So I was thinking, if there's like a more coarse-grained, more discrete way, would you observe the similar pattern that we're seeing right now?
Well, we wanted to incorporate the electrical properties, right? Because that was something that is, foundational part of Hodgkin-Huxley, and part basically which can be traced back to, some foundational math from ages ago. We wanted to be able to capture that. As it turns out, we're running these on NVIDIA Grace Hopper GPUs, which are like on our UVA campus. This is a very special cluster just for neuroscience, and these GPUs are 400 times faster than whatever else we have. So we're not too worried about how computationally expensive it is. And we've also optimized this to run pretty quickly. But in terms of taking into account those electrical parameters, we thought they were very important to have been included.
Because, like, in my head, I'm imagining, like, for example, you're saying they're like different variation of the speed according to like different layers and stuff.
Yeah.
So I'm thinking, like, if the degrees of like connectivity, like two things connect to like one node, there's like going to be a race condition and stuff like that. It'll be pretty hard to do it from, like, if you're being like 100% loyal to the Ohm's law you're talking about, right? So how did you guys like just?
I'm not sure what the alternative would be if I'm not 100% committed to Ohm's law.
Oh yeah. I'm like, I'm just like thinking, oh, like we're talking like variation of like neuron speed here. So maybe just like assign them for like a number or something, and then they arrive at a discrete time, and then there's going to be a queue, and then you can determine which race condition you're going to deal with this for each of the neurons. So
one thing we are doing, I don't know if this gets a direct answer to your question, but we can reconfigure the wiring however we want. So we could actually have, wiring actually much more extensive if we wanted. It's not a fully connected everything to everything system. So things are just very strategically connected. We can manipulate that, but we've arrived at this sort of out of parsimony, right? We wanted to keep it relatively simple. Every node is not connected within a layer, isn't connected to every other node in the layer. It's just connected to its immediate neighbors. So that basically does message passing, right? So you could imagine, like, if it's the wave at a sporting event, if somebody stands up over here and everybody's in on the joke, right? Somebody stands up over here, the next person stands up, the next person stands up. So you get that wave. If you start making it everything to everything connected, you won't really capture that quite right. Now, the brain's somewhere in the middle, so we sort of assumed a least connected kind of a structure, but it's still pretty connected, if that makes any sense. How that actually maps to Ohm's law and race conditions, we have the ability to go and test alternatives, so we could see whether or not there's any downside to doing it a different way.
I see. Okay, thank you so much.
Yeah, thank you.
Other questions? Yep.
I feel like I have a very basic question.
Okay.
I'm lost as to how learning is happening or could happen in this model.
So what we're doing is we're going to be using like a mutual information based thing. So if you presented a 1 and you have it kind of like, say, based on the spectral characteristics at the output nodes, we're going to label that as a 1. We're just basically going to count those up, and basically do a mutual information on what we stuck in with what the organoid thinks it is. That's going to be fed back into making little adjustments to the distances, but the signals have to travel, modulating that G ratio, and maybe controlling some other things as well. So that's probably what I think we're going to be done with by next week. But I didn't want to present it too early, because then it wouldn't work and I'd come back in a year and you guys would laugh at me. I don't want that.
Is the G ratio what is usually the weights between neurons and a typical neural network? Is that the analogous
It's
parameter that we are tuning here?
It's a parameter that you can tune. There are theories that the myelination is changing in very small levels, especially in development, where signals are learning their best path, and if they're a longer connection, that myelination gets kind of incorporated. As you age, you can get demyelinating diseases that can slow down the system. So being able to train something and say, older organoid versus a younger one, maybe also something that we could like explore. And maybe that'll give us some insight into, what happens in demyelinating diseases, what happens in Parkinson's disease. Again, small scale, very simple model, but we're hoping for big insights.
Is there another question? Yeah.
Yes.
Is there any interest in using these digital organoids to simulate like neurodevelopment or just intermediate stages of neurodevelopment, or are you focusing primarily on the mature brain?
No, I kind of think that they actually are a really good model for development because, again, we'll show you a little bit more about this, in the future, is as they change their shape, it's sort of like going through a developmental process, right? And you're altering the structure of this. And again, the whole idea is to make sure that signals get to where they need to go as quickly as they need to go, as efficiently as possible, given these geometric constraints. The brain develops in a lot along those similar lines, that you've got a skull you have to fit inside. Our brains aren't this big, right? That you've got some geometric constraints. You need to make sure that signals are getting from one part of your brain to another part of the brain as efficiently as possible, right? There may be multiple pathways, so you may have multiple routes to go. This gives you some plasticity. It gives you redundancy. But if you lesion one, you may be, signals may be taking a slower path. And that may be something that happens in stroke or in traumatic brain injury or TBI, that sort of thing. So there's a potential to look at development as well as lesioning to simulate what happens in disease.
Oh, thanks for the great talk today. How is it essential that your digital organoids three-dimensional as opposed to two-dimensional?
Or four.
Or four. Yeah, let's say two. So did you have any reason you wanted to make it three-dimensional?
We stopped at 3 because if you take just kind of the left to right neural network and you wrap it around a circle and create a disk, that gives you one format. But if you go this way, you basically get a sphere. And we just thought that would be a good thing to stop with, right? So it's three-dimensional and we're playing with time, largely because organoids are three-dimensional, but they have, activities or processes which play out over time. So this is kind of where we did it. I presume that if you could figure out how to embed it in an even more high-dimensional space, that you could do it that way too.
I'm interested in the other direction. So I think
I see.
You can do lots of things. You present it with a two-dimensional model, and also the brain is sort of two-dimensional, right? So if you look at the gray matter is 2.5 dimension rather than 3.
Yeah.
So
we did it so that we could get that three-dimensional traveling wave, so that, say, signals are traveling into the organoid as well as spreading outward,
sure.
around the different layers. So we're trying to take advantage of the spreading of the waves within a layer. That's going to be your two-dimensional process, but it's also going into the center of the organoid where we have that little output node structure that catches the signals. So those are the justifications for the 3 dimensions. Sure. It could be embedded in a higher manifold space, or you could model it on 2D as well, but you wouldn't fully capture that traveling wave dynamic as nicely.
Makes sense.
Yeah, thank you. Good question.
May I suggest that we end here? Dr. Marconi is going to be here for another few minutes.
Yes.
Okay,
If anyone wants to come up. Thank you, everyone. I've really enjoyed my visit. Thank you.